Hi, everybody. Welcome to the last day of Neuromatch. I hope you've been enjoying the meeting. I'm Robert Froemke at NYU, and it's my pleasure to introduce Ioana Carcea from Rutgers here to give a talk today. So Ioana is an MD PhD, and she did her PhD work at Mount Sinai in Deanna Benson's lab. And then she did a postdoc in my lab. And I sort of lost count. I just looked this up on PubMed. Ioana and I wrote a total of 10 papers together and counting. And what she's gonna be talking about today is paper number 11. She's had her own lab for about a year now or so. So take it away, Ioana.
Okay, sounds great. Thank you, Rob. Let me share my slides. Okay, this is good, right? You can see them? Okay. So hello, everybody. Thanks for being here. Today, as well, Rob didn't mention the title, but I will be talking to you about our work on how maternal behavior can be socially transmitted in mice. We all know that maternal behavior is essential for survival of the majority of mammalian and avian species. And it is such an important behavior, in fact, that in many cases, unrelated conspecifics contribute to caring for the young. These individuals are called alloparents or non-biological parents. And they have been described in more than 130 different species, including primates, whales, dogs, and rodents. Rodents, and in particular mice, are a great model to study how alloparenting behavior develops. Mice in the wild live in colonies, generally composed of one male, a couple of reproductive females, and several non-reproductive females, who are, in general, daughters of the colony that stay around to help caring for new generations of pups. So how can we best measure mouse maternal skills in the lab? One robust test for this is the pup retrieval behavior. So here, what we do, we isolate a pup from the nest. And when isolated, this pup will start emitting distress calls in the ultrasonic range, like the example call I'm showing you here. When hearing these calls, the mother immediately, within seconds, locates the pup and picks it up, retrieves it back to the nest. It has been shown previously that this behavioral response depends on the activity of the left auditory cortex, where these pup vocalizations are primarily processed. Importantly, virgin females that have experience with pups can also retrieve pups. And when oxytocin, a hormone which we know to be heavily released in mothers during parturition and lactation, when this hormone is given to virgins, either systemically or topically in the auditory cortex, the left auditory cortex, these animals can also start retrieving pups. On the contrary, inexperienced virgins or animals where the oxytocin activity has been decreased do not retrieve pups. So what we see here is that experience and oxytocin go hand in hand when it comes to pup retrieval behavior and then more generally to maternal behavior. So we wondered then if experience could use the endogenous oxytocin system to induce alloparenting behavior in virgin female mice. Before I go into how we address this question, I want to tell you a little bit more about what I mean by experience. Traditionally, virgins are allowed to gain experience with pups by co-housing them with a number of pups for hours or days. More recently, we started co-housing the virgins not just with pups, but also with the mother of those pups. So one of the first things we did is to test how these two different types of experience can impact alloparenting skills in the virgin, and in particular pup retrieval skills. So here I'm showing you a graph where we plotted the probability of virgin retrieving on the y-axis, and on the x-axis is the time in days of cohousing. What you can see here is the probability that virgins retrieve after being cohoused just with pups. And you can see that they do start retrieving pups. However, when we cohouse virgins with the dam and pups, we see that the acquisition of this pup retrieval behavior is significantly accelerated. So virgins start retrieving much sooner. And the question then is, how is this socially mediated acquisition of pup retrieval behavior induced? What exactly happens during this cohousing experience? So to answer that, we took three approaches. One was to look at behavior, particularly or specifically interactions between the virgin and the dam. Secondly, we wanted to understand how these interactions might modulate the activity of the oxytocin system, which is the main neuromodulator that we know to be involved in maternal behavior. And lastly, we wanted to understand if this cohousing interactions might change the representation of pup calls in the auditory cortex. So to answer this, we built a system for continuously monitoring the interaction between animals throughout the cohousing procedure. So the system has a camera that acquires continuous video recordings, has an ultrasonic microphone for picking up pup calls, and a neural activity acquisition system for recording the activity of oxytocin neurons in the virgin hypothalamus. So with this system, we acquired days of video recordings, and we first analyzed those to look for interactions between them and virgins. And we indeed identified two behaviors that I will describe, which we believe to be important in the acquisition of alloparenting skills by the virgin. So the first one of these two behaviors is the fact that the dams direct the virgins towards the nest. So I'm going to show you an example in this video, and what you'll see here is the nest with pups. The dam, you can see, is already emerging out of the nest, and she's headed toward the virgin, who is in a different location of the cage. And then you'll see the mother directing the virgin towards the nest. So the mom comes out, and then she pushes the virgin towards the nest. She will join the virgin in the nest subsequently. We see this kind of spontaneous interaction occurring many, many times during the cohousing procedure. And we wanted to look at the spatial organization of this interaction. So here I'm showing you a representation of the cohousing enclosure. The white circle represents the location of the nest, and the yellow pixels represent the probability that the mother will start chasing the virgin at that location. As you can see, the start points are, the vast majority of them are outside of the nest. However, when we look at where this chasing ends, we see that almost exclusively the chasing ends within the nest. So we find that this chasing that's directional from outside of the nest towards the nest, which we call shepherding behavior, occurs about 85% or more of all chasing done by mother. So what are the behavioral effects of shepherding? What might they be? Well, obviously one of them would be that the virgin ends up spending more time in the nest. And that's indeed what we see when we quantify the percent of time spent in the nest by the virgin over the course of cohousing. But importantly, we also see something quite unexpected, and that is the fact that the frequency of shepherding during the first day of cohousing correlates with the probability that the virgin will start retrieving pups at the end of the first day of co-housing. And this is a very strong correlation. So this suggests to us that the more the virgin is shepherded, the more likely she is to retrieve pups consequently. The other behavior that, before I go into the other behavior, just to summarize the fact that we see the shepherding occurring many times throughout the co-housing procedure. I didn't show you, but it depends on the presence of pups in the nest. And I showed you data indicating that shepherding contributes to the acquisition of alloparenting behavior in virgin. So the other behavior that we found is that the dams self-generate maternal retrievals during co-housing. You will see in this example video that I'll show that sometimes when the mothers come out of the nest, they drag accidentally one or more pups with them, and then they return to retrieve those pups. So you saw the pup coming out, being dragged out by the mother, and then the mother returns to retrieve it back to the nest. And the virgin is present in the nest and has the ability or the capacity to observe what the mother is doing. So we wondered, given that the virgin has this possibility repeatedly during co-housing, is it possible that virgins can learn pup retrieval by observing the mothers doing this behavior? So to answer that, we first wondered if there is a correlation between the frequency of dam retrieval during the first day of co-housing and the probability that the virgins retrieve at the end of that day. And again, we find the correlation here is not as robust as the correlation with shepherding, but it exists. Furthermore, we then isolated this interaction from the context of the co-housing behavior. And so now we repeatedly exposed virgins to maternal retrievals in series of 10 for a total of 4 days. So each day the virgins observe 10 maternal retrievals, then they get a break, and then they are tested themselves for pup retrieval. So what I'm showing you here are 15 virgins that have been exposed to this paradigm. And in green, you see the probability that they will start retrieving pups. As you can see here, the vast majority of virgins start retrieving pups sooner or later during this procedure. Now, we wondered what might be the sensory modalities that contribute to the acquisition of pup retrieval by observation, by being exposed to dam retrievals. So to start answering that, we first wanted to block the touch interactions between the virgin and pups and also between virgin and dam. So to do that, we placed a transparent barrier in the cage that allowed visual, auditory, and olfactory information to still cross across this barrier. So when we do this, we see again that many of the virgins, again, the majority of them, start retrieving pups after being exposed to maternal retrievals via the transparent barrier. However, if we make this barrier opaque, blocking visual information, now we no longer see robust acquisition of pup retrieval behavior by virgins. In fact, only 2 out of 18 animals showed pup retrieval behavior after the opaque barrier condition. Yes. So this indicates that there is a visually guided observational learning of pup retrieval behavior that might occur during co-housing. So I showed you that both shepherding and observational learning might be behavior interactions, spontaneous interactions between adult animals that allow the virgin to behave as an alloparent. And now what we wanted to address next is whether these behaviors can use neuromodulators, in particular the oxytocin neurons, in order to contribute to the acquisition of pup retrieval behavior. So to start testing that, we first wanted to know whether the activity of oxytocin neurons in the paraventricular nucleus of the hypothalamus is required for acquisition of PAP retrieval during cohousing. We focused on the PVN, the paraventricular nucleus, because this is where oxytocin neurons that project centrally to other brain regions are located. So what we did here, we induced the transduction of an inhibitory dread, specifically in oxytocin neurons. And then we administered CNO to these animals, to the injected animals, over the course of cohousing. And we found that in this CNO condition, there is a decrease in the acquisition of PAP retrieval behavior compared to control conditions. So this indicates that the firing of oxytocin neurons in PVN is required for acquisition of PAP retrieval behavior during cohousing. Now, if shepherding and observation of dam retrievals are indeed important for acquisition of PAP retrieval behavior, then we would expect that they are able to activate oxytocin neurons based on this result. To test this, we decided to do single unit recordings in the PVN. We implanted an electrode bundle in this very small structure, and we used co-registered MRI and computer tomograph scanning to verify the proper location, the correct location of these electrodes. Moreover, to specifically record the activity of oxytocin neurons, we induced the transduction of channelrhodopsin in these cells, and then we used blue light to determine which neurons respond robustly and with short latency to this blue light laser. So those neurons that respond in this way are going to be bona fide oxytocin neurons, and the rest of the neurons we're going to name unidentified PVN neurons. So let's look at how these neurons respond to the behaviors I described earlier. Here's a cell recorded in PVN where the spike trains have been aligned to the onset of shepherding. You can see the different shepherding episodes highlighted here in red. And as you would expect from a spontaneous behavior, they have different lengths and they sometimes occur with different frequencies, sometimes closer to each other, sometimes further apart. We looked at the average firing rate during the shepherding events and compared that to the firing rate during baseline, during the preceding baseline. And what we see is that this particular neuron is strongly activated during shepherding behavior. Overall, when we look at all neurons that we recorded, we find that 19% of oxytocin, bona fide oxytocin cells, respond to shepherding by being activated by this. And by comparison, only 12% of unidentified PVN neurons are activated by shepherding. Interestingly, neurons that do respond to shepherding only respond to this type of event and not to another event that is similar in terms of motor characteristics, but not dissimilar in terms of the social motivation. So here we see that neurons that respond to shepherding do not respond to events when the virgin is pursuing or chasing the mother. As I mentioned earlier, shepherding is followed by the virgin entering the nest. So we also wanted to see how oxytocin and other PVN neurons respond to nest entry. Here I'm showing you 9 units recorded simultaneously in PVN. One of them is a bona fide oxytocin cell in blue here. And what we can see is that several of these neurons, including the oxytocin neuron, has increased firing after the virgin enters the nest. And that is more clearly shown in this peristimulus time histogram here, histograms for example cells. So overall, we find that a similar number of oxytocin and unidentified PVN neurons respond to nest entry. About 33% of them are activated by nest entry. Importantly, this happens when the nest entry follows shepherding, but also when the nest entry is voluntary. So this indicates that there might be cues related to the nest, either visual, tactile, olfactory, that generate a response of oxytocin and other PVN cells. How about observation of dam retrievals? Here I'm showing you 4 units that again were recorded simultaneously in virgin PVN while the mother retrieved a pup during cohousing. The spike trains are aligned to the onset of the retrieval event, and you see that preceding that we have an interval where the pup vocalizes. It emits distress vocalizations. And what we can see here is that, again, the oxytocin neuron in blue here and some other PVN neurons respond to the onset of dam retrieval. So here I'm showing you that almost 30% of oxytocin cells are activated by observation of dam retrieval, and by comparison, only 17% of unidentified PVN neurons respond to this behavior. So I described these 2 spontaneous interactions, shepherding and observation of dam retrievals, and then we showed that both of these are capable of increasing the activity of oxytocin neurons in PVN. I should mention here, although I didn't show the data, that we have control behaviors that occur during cohousing, which we don't find to activate oxytocin or other PVN neurons. For example, entry into an empty nest is not capable of activating the cells to the same extent as entry into a nest with pups. All right, so then let's see what are the effects of oxytocin neuron activity and PVN neuron activity on the processing of pup calls in the auditory cortex. Previous work in Rob Froemke's lab showed that there is a really striking difference in how the auditory cortex of mothers and virgins, inexperienced virgins, process pup calls. So here we see a very robust response in the auditory cortex of mothers in response to playback pup calls. By comparison, the virgin auditory cortex does not robustly respond to playback pup calls. This increased responsiveness of the auditory cortex is important for pup retrieval. When the auditory cortex is silenced using mucimol, we see that there is a decrease in the success of pup retrieval behavior compared to pre- and post-activation conditions. More recently, Jennifer Schiavo, also in Rob's lab, showed that there is a transition from low responsiveness to high responsiveness in the auditory cortex of virgins during the cohousing behavior. So we wondered if this transition is mediated, at least in part, by activity of oxytocin and other PVN neurons, then we should expect to see a correlation between neuronal activity in the 2 structures. And indeed, this is what we observe here. I'm showing you dual recordings in the auditory cortex, where we did fiber photometry recordings, and in PVN, where we did single unit recordings. And we did this dual recordings during the observational learning across a transparent barrier. And what we can see here is that there is a correlation between the activity in PVN and the activity in the auditory cortex, which is robust and significant, as you can see here. Now, given this, we then wanted to know if blocking the activity of oxytocin in the auditory cortex can impair pulp retrieval behavior or the acquisition of pulp retrieval behavior by observation. So again, here we did the observational learning with a transparent barrier, either with animals where we injected saline in the auditory cortex or animals where we injected an antagonist for the oxytocin receptor in the auditory cortex. So again, not again, but importantly, compared to control conditions, we see that blocking the oxytocin receptor impairs the acquisition of pulp retrieval behavior by observation. So overall, I showed you that the spontaneous behaviors that we identified during co-housing can engage the endogenous oxytocin neurons in the PVN in virgin females, and that this activity might be important for controlling the representation of pulp calls in the left auditory cortex of virgins. So with this, I would like to thank my lab and also many people in Rob's lab. I'm just mentioning some of them here, but there's many more that contributed to this project. Also collaborators at Rutgers, where I am now, and at other institutions. I would like to highlight John Cunningham, with whom we're collaborating now on automating the analysis of video recordings, and Justin Reisberg from Mount Sinai, who's done a lot of work on the neural analysis and continues to help us with that. Okay, thank you. And I will take questions now.
Okay, great, thank you. So we have several questions. Have you tested if virgins that didn't retrieve pups with the opaque barrier testing then would retrieve pups with the transparent barrier?
So basically use the same virgins in the two conditions. We haven't done that. One thing that might speak to this experiment, should I stop sharing? No, I'll keep going. So one experiment that we do that could speak to this is we are now currently doing an experiment where we activate oxytocin fibers that project to the auditory cortex during the opaque barrier condition experiment. And so basically what we're trying to see, if we artificially induce the activity of oxytocin neurons, particular oxytocin fibers in the auditory cortex, during this paradigm that generally is not effective, can we then increase the possibility that these virgins learn to acquire pulp retrieval? And that's what our preliminary data shows. In fact, out of all virgins that we tested so far, all of them learned to retrieve pups with the opaque barrier as long as we activate oxytocin fibers in the auditory cortex.
And what about blind virgins? Do you think blind virgins, what mechanisms do you think they have to learn to retrieve?
Right. We haven't done that. I mean, it's a little bit tricky, and some decisions would have to be made as to when we want to blind these animals, right? Do we want to blind them immediately after birth so that they never learn to rely on visual cues, or do we blind them as adults, in which case they might behave very, very unpredictable, given that one of their sensory modalities is all of a sudden gone. We don't even know what to expect from their, motor activity, exploratory behavior in the cage very much. So we haven't done those experiments. I think it would be good to do them as long as we can figure out the exact timing for when we should blind these animals.
So about the oxytocin neuron recordings, what are your criteria to define if a neuron is being activated or suppressed?
Activated or suppressed. Yes. So here we, calculate the average firing, or for each trial, we calculated the firing rate during the event and compared that to the baseline. And we do permutation tests to see, if we were to move these events in time, would we be would this difference in firing rate be above, 95% of what we would get with all permutations? So that would define activation, and inactivation or suppression would be lower than 5%.
How homogeneous or heterogeneous is the oxytocin neuron population? Is it the same population that's activated during shepherding and nest entry, or are they different populations?
We do have the data. There is some overlap, but not much. So it's interesting, and it seems to be the case that it's mostly different populations that respond to probably different cues, right? We would expect that shepherding, nest entry, observation would all have different or rely on different modalities to reach the oxytocin neuron population. And we do see that they're largely non-overlapping.
So you showed data on the dread-based suppression. But what about after animals have already learned? If you inhibit or block oxytocin neuron firing, does that impair execution of pup retrieval?
We did that experiment as well in virgins that already acquired the behavior, and we don't see the same effect with chemogenetics. Now, there might be a trend, but it's mild. Now, I don't know, perhaps in mothers that would be different, perhaps with more animals this would be different, but we didn't find a significant difference in virgins that already acquired the behavior.
Follow-up question to the question about blind animals and the need for visual input. Mm-hmm. Basically, can you clarify how you're thinking about it, what the hypothesis is? Is visual input required for oxytocin release, even in auditory cortex? And oxytocin plus the sounds of pup calls then required for successful retrieval, either behaviorally or retrieval learning initially?
Yeah, so the way I think of this in the context of the observational learning paradigm, I do think that the visual input is required for increasing the activity of oxytocin neurons that will result presumably in the release of oxytocin in the auditory cortex. And that in turn will induce a stronger representation for pup calls in this brain region, which is necessary for pup retrieval. So yes, I think this would be the mechanism pretty much in the case of the observational learning. And, again, I would say that the fact that we can replace the visual input just with activating oxytocin neurons, oxytocin fibers, I think it's further proof that this would be, if not the mechanism, at least one of the mechanisms. Now, of course, the mechanism, as I said earlier, would be different when we think of oxytocin cells being activated by shepherding or by nest entry. But yeah, in the case of observational learning, I do think the visual input is key.
Maybe on that note, you could talk a little bit more about the specific aspect of the visual input. Oxytocin clearly enhances sensory responses to pup cues, like pup calls. But what about potential imitative parts of learning? If, for example, do you think there are changes or activity elsewhere, for example, in M2, higher motor cortex areas that are also important?
I think this is a fascinating question, and we haven't looked at that. I think we should, in fact, look at that aspect. But in terms of how much this is imitation versus, object enhancement or location enhancement, or, some people might go as far as emulation. We don't know that. I mean, imitation is probably harder to argue, right? Because each pop retrieval is by design going to involve a different motor sequence, right? Like the location of the pipe is not going to be precisely the same for every trial. The position of the adult animal is not going to be the same for each trial. That's up to them where they want to be in the cage. So saying that this could be imitation is probably not, in the stricter sense, is probably not likely to be imitation. Yeah, just because we do not see the same exact motor sequence in the mother and the virgin. Now, having said that, I don't think we should, exclude the possibility that the motor cortex is involved in this. In fact, I do believe that plasticity in the motor cortex is probably a big component of this behavior as well, right? I mentioned, I think in the beginning, that mothers very quickly detect the isolated pup and move swiftly towards it, pick it up without a problem. Now, like, the virgin doesn't do that. The virgin takes a winding path to get to the pup, if she goes there at all. Then we have some studies on errors, and we find that they initially grab the pups in weird, different ways. They grab it a little bit and then drop it, and they're very awkward in their motor skills, and that refines over time. They become experts over a relatively short period of time. Okay, so I do think there is plasticity in the motor cortex, and I do believe that we should look at that as well. But I would not necessarily expect to see, the classical motor mirror neurons or anything like that, just because the paradigm is not designed to show those neurons or to highlight those neurons. I didn't mention this talk, but we do see sort of mirror neurons in PVNs. So neurons that are activated by both observing them retrieving and by the virgin executing that retrieval. It's not the exact sequence of activity, but the same neurons are activated by both of these. So in that sense, there is sort of like a social maternal cell population in PVN, or if you want, mirror maternal neurons in PVN.
Okay, have you looked at markers of plasticity in oxytocin neurons over time? Maybe CFOS or other things might indicate a change in the system.
Over time, like during co-housing or during the, we are now using the TREP2 animals to look at FOS activity in different behavior settings. We don't have yet an answer on that. But bottom line, no, we didn't do much of that. I believe actually Silvana in your lab, Rob, is working on something related to plasticity markers. So she would have a better answer to that, but I haven't looked.